Hello student, I am going to help you by this video about the examination which is near about on 4th we have a science paper. So how can you proceed in biology section or even in science? On the board you are observing most of the students they have a problem in Mendel's law of heredity. So Mendel's observed through his experiment and explained the three law of dominant, law of segregation, law of independent assortment. Questions may be asked in different way, but your answer will be like that. You have to describe the monohybrid cross. How can you describe? Students generally commit the mistake. So please don't take it in an ordinary way. This is your board examination. So kindly, what suggestion is given to you, you will write the answer accordingly. Here, I have made a monohybrid cross. Suppose that question is asked that when a tall plant is crossed with a dwarf plant, in F1 generation, all plants appear to be tall. When they were self, then in F2 generation, some dwarf plants also reappear. So, what is the mechanism behind it? How can you explain this? So, try to understand. Before describing any type of question in monohybrid cross, first of all, know what is the monohybrid cross? When one pair of contrasting traits, that suppose that tall and dwarf, I am crossing with tall and dwarf, the cross is said to be monohybrid. So, one pair of contrasting trait is considered during the cross, during the hybridization, taking male anther from one plant and ovum from other, that is called crossing or hybridization. This sign indicates is known as monohybrid cross. So you can write here capital T, small t, small t. Homozygous tall and here homozygous dwarf. Homozygous means all the factors are identical. Both the factors are identical. The first postulates given by the Mendel is that character is governed by factor which exist in pair. That's why I have written in pair. One given by one parents, another given by another parents. Means father and mother. So suppose that this plant, this represents the plant. Parental generation is the plant. Having the one factor come from its male parents, another from female parents. Similarly to this one. So when they form their further during reproduction, they form their gamete. So gametes are haploid. Parents are diploid. Gametes are haploid. So during haploid formation, only out of the two capital T, one capital T will come here, one small t will come here. So these are the gametes. Gametes are in circle. You commit this mistake, you student. Okay? So don't commit this mistake. Gametes should be circle. Don't leave the gap of gametes. Write the parental generation, then gametes, encircle it, and these gametes unite together. So by this union that you are uniting them to in F1 generation. So this one is F1 generation. F1 generation was also appear to be tall, similar to the one of the parental type. We unlike our expectation that it should be intermediate type, but it was found to be total tall, like the parent one, dominant one. So when this F1 was self, F1 selfing, when we write here selfing, here already written. Selfing means we take the pollen grain from the same flower and the ovary and the ovule and egg of the same flower. So you know the plants are the flowers. Majority of the plants, the flowers are bisexual. So we can write the here easily. Heterozygous, but the tall, though plant is tall, but it is heterozygous. One capital T, then another small t. Now, you have to understand here that a small t doesn't express itself. It doesn't mean that a small t is absent. A small t is present, but in the presence of dominant factor capital T, a small t unable to express itself. But when there is a chance given to it in the next generation, in F2 generation, we write the F2 gametes. Again, the students commit mistake, they don't write here male and female. Suppose that this one is a male gamete, capital T, a small t, from male side. Similarly, from female side, a capital T, a small t. Probability, any male gametes align with the female gametes, any female gametes. So these are the probability we can express in through Punnett square. So capital T, capital T, capital T, a small t, again capital T, a small t, and a small t, a small t. So these are written the genotype. These are the genotype. The students also do the mistake. While solving this question, they make in a small area, no need. Take a big page, okay, whole page is utilized, write the genotype as well as the below genotype, you must write phenotype too. Genotype means genetic constitution. What the capital T and capital T small t, both are taught. So phenotype is the same, morphological observable feature is the same, but genetic constitution, genetic combinations are different. So that is genotype, what is inside and what is looking outside morphologically is the phenotype. So here you can write, and at last when we write the phenotype, the ratio of phenotype, again students commit this mistake, again I am telling you, phenotype ratio of F2 generation, these are the F2 generation, the probable gametes of F1 is sort together to give rise the F2 generation. So F2 phenotype ratio of F2 generation in which type of cross? Monohybrid cross. When you take dihybrid cross, the ratio will change. So you must write this one. You forget this one. Many of the students do this mistake. So what is this? Monohybrid cross, what is the ratio? Tall is to dark. Don't forget to write whatever be the character, whatever be the traits given. So you write here tall and dwarf. So when we count 3 tall and 1 dwarf, so 3 is to 1. When we write the genotype ratio, question will be asked, derive the genotype and phenotype ratio. So you will write here that genotype capital T 1, capital T a small t 1 and 2, and a small t 1. So 1 is to 2 is to 1. So total genotype when we write, so capital T 1, capital T small t 2, small t 1. So genotype ratio is 1 is to 2 is to 1 in monohybrid cross. Now you can explain the law of dominance on the basis of this cross. First of all, you draw the cross and then you describe that what's the Mendel's first postulates or first law of dominance. So Mendel, on that observation, you can explain that Mendel's law of dominance as that in a monohybrid cross, the F1 generation, in F1 generation, only one factor expresses itself masking the other. The factor which expresses itself, like the capital T, tallness, is the dominant, which expresses itself masking the small, that is the dwarf or dwarf. So here the tallness is the dominant. So Mendel explained that either of the two traits, one is dominant. One is able to express in heterozygous condition, capital T small t, in capital T small t condition, which expresses itself, capital T. Hence it is a dominant factor, and this law is known as law of dominance. Right? Stated. But it doesn't mean that the recessive factor has gone. No, recessive factor is still there. So in F2 generation, when there is no dominant factor, consider a small t, the small t able to express itself. So without blending, without mixing with capital T, the factor which decides the character pass on generation after generation without blending. But out of the pair factor, one dominating the other. So this is law of dominance. Law of segregation you can also explain that in parental generation factor exists in pair. Like here, capital T small t, capital T, small t. But during gamete formation, when we write the gamete, when we write the F1 gamete, we write what? F1 gamete, capital T and small t separately. So in parental generation, parental generation, factor exists in pair, but during gamete formation, only one factor goes. This one is another, this one is another gamete. So gametes are the pure for the traits. Gametes contain only one factor, not the double factor. This is the reason that always when you write the gametes, you must circle it. That's so that you are write the haploid set. So in this way, Mendel's law of dominance, law of segregation, you can easily answer this question. Okay, I am giving you some other point too. How can you proceed? When I explain detail, it will take too long time. But some important questions I am giving to you, you must remember, and as well as the suggestion, the point which is to be revised during this time. First of all, what is nutrition? Revise. What is nutrition? What is autotrophic nutrition? The equation of photosynthesis, you must know that how the inorganic substances unite together. Photo means light, synthesis means put them together. So carbon dioxide and water combined in the presence of sunlight by chlorophyll bearing plant to produce glucose. Oxygen is a byproduct and in the presence of sunlight. So this is photosynthesis. So photosynthesis question may be there along with the photosynthesis. Other question may be asked like saprophytic, symbiotic. In saprophytic, that organism obtain their food material from dead and decaying material, digesting, oozing enzyme outside, digesting them, then absorb. In heterotrophic mode of nutrition, amoeba, nutrition in amoeba may be question asked. Maybe this question, this type of diagram may be asked. You have to label that if cuticle protect is from dying, and then there are mesophyll cells which are involved in photosynthesis. The lower surface of the leaf bear the stomata. So here is the stoma, one plural is the stomata. So in this type of question, chlorophyll, this is the diagram of chloroplast. So it has a granum region and a stoma region. Granum region is the site of light reaction. Stoma is a site of dark reaction. You must also remember the diagram of stomata. You can see in your NCERT book also it is given that the pore is called stomata. Here the pore is called stomata. These are the guard cell. This one is the nucleus. These are the chloroplast. The outer, the inner wall is thicker and outer wall is thinner. So in this way, turgid stomata. When the stomata means the guard cell turgid, the stomata opens. So in light reaction or dark reaction, when we explain this in the light or model of light and dark reaction, so what is happening in light reaction? In light reaction, the photolysis is taking place. You can observe here that water is splitting into oxygen and hydrogen. Hydrogen is accepted by NADPH2. So NADPH2 formation, ATP synthesis and evolution of oxygen taking place in light reaction. So what the assimilatory power, that is NADPH2, nicotinamide adenine dinucleotide hydrogen phosphate and ATP are assimilatory power which are used in C3 cycle, that is the dark reaction site is the stoma. So grana perform the light reaction, stoma perform the dark reaction. Nothing more than that. This type of question will be asked to you. Now, maybe question asked that what are the factors which affect the photosynthesis? So carbon dioxide concentration, water is there, sunlight is there, temperature is there. You can easily write, describe this one factor. Now, experimental question may be asked that starch is produced during photosynthesis. Easily you can do it experimentally if you have done the practical examination, you are already familiar with. So test for the starch, or if it is asked that chlorophyll is essential for photosynthesis, don't forget to write the variegated or coleus or croton plant having the variegated, means somewhere the leaf is green and somewhere non-green. Then we make the design of the green and non-green part and then describe accordingly requirement, procedure, observation, conclusion and inference along with the precautions. So experimental question you can easily describe. Nutrition in amoeba. In fourth step you can describe the nutrition in amoeba, that pseudopodia. When amoeba sense that there is a food, it start developing the pseudopodia, is a locomotory organ as well as the food capturing organ. Now the food is nearly to be captured by pseudopodia. The food cup is produced. When lysosome encountered with the food cup, it forms food vacuole. As soon as the lysosome encountered with the food cup, the digestion begins. With lysosome contain hydrolytic enzyme, so digestion begins, and amoeba digested. After digestion, assimilate the food inside the body, and waste material is ingested when it's come to the surface of the body. So you can explain, as well as two marks question is generally asked related to this one. In human digestive system, you must practice about your drawing the diagram, that from mouth you can say how can the stomach, and then small intestine, large intestine, rectum. So you can write easily. And question may be asked that what is the role of gallbladder? Store the bile, so bile scent increases the pH. In stomach, the digestion taking place in acidic medium. The food which is chewed by the different type of teeth, we have a dental formula 2 1 2 3, so you can remember this one. Pancreas secreted the enzymes, carbohydrates, protease, and lipase, so digest the food in the first part, that is the duodenum part of the small intestine. And pancreas itself is a gland which secretes enzyme, mixocrine gland. It also secretes the insulin which remove the blood sugar. So question may be asked like this. So what are the steps in holozoic mode of nutrition? You know that ingestion, digestion, absorption, assimilation, and ingestion taking place. What is digestion? Question may be asked. Enzymatically control hydrolytic process, where complex food molecules are broken down into simpler ones, soluble one, is known as digestion. So you can easily write. And then question may be asked related to the absorption. So how your small intestine, the microvilli are finger-like projection. They are numerous in number, increases surface area, highly blood vascularized, so best suited for the absorption of food. The fat is absorbed by the lacteal, different type of part, and the glucose and other amino acids are digested, absorbed by the blood vessels. So in this question may be asked to you, so you must be prepared to answer this one. In respiration, we have an aerobic and anaerobic form of respiration. You will revise easily, you can do. So the root of the aerobic respiration and anaerobic respiration, how the glucose converted into pyruvic acid, then into aerobically lactic acid, and anaerobically lactic acid, and ethyl alcohol, anaerobically carbon dioxide and water. High amount of energy is produced. Examples you can write. Then the yeast perform the anaerobic alcoholic fermentation or anaerobic respiration. In our muscles, lactic acid produced, and aerobically when pyruvic acid, which is the 3 carbon containing compound, formed during glycolysis. Lysis means breakdown, glycose means glucose, so that is used in aerobic respiration. In respiratory mechanism, breathing mechanism may be asked, that diaphragm, intercostal muscles contract, that increases the lungs volumes, and when the lungs volume increases, the air pressure decreases, outside the air rush towards the body, and that is called inhalation. And just vice versa occurs in during exhalation. So gaseous sections, you can, by the process of diffusion, gaseous sections takes place from the lungs to tissue, and then tissue to again lungs. So question may be asked like this. In transportation system, you know how to draw the diagram of heart. Okay, so just make a 6 opposite 6 like a structure, and give a partition, and then left or right. So right part receive the blood, give 2 opening. Remember the diagram, I have already explained to you, that from right side, inferior vena cava, superior vena cava, blood come from different part of the body, then come to the right ventricle, then right ventricle. From right ventricle, the blood goes to the lungs through pulmonary arteries, and then from lungs it is oxygenated, then return to the return by the via the pulmonary veins into the left atrium, and then left atrium to left ventricle, and from left ventricle it goes to different part of the body. So in this way you can also show the circulation of blood. So your circulatory system comprises what? Heart itself, the blood vessels, and the blood itself. So heart is a pump organ. The morphology and function of heart hardly asked, but morphology generally and the path is generally asked. Difference between arteries and vein is very important. The arteries are deep-seated, you can see here. Arteries are deep-seated, veins are superficial one. Arteries contain oxygenated blood from heart to different part of the body. Vein return the blood from different part of the body to heart. So in this way you can easily describe this one. And lumen of arteries is narrower and lumen of vein is wider. So in this way you can capillary coordinate between the veins and arteries. So a separate video I have made for this one, you can see if you have problem. In heart you can easily draw this diagram, no problem. 4 chamber, the upper one are atrium, lower one are ventricles. Right side always contain the deoxygenated blood, left side contain oxygenated blood. So we have a double circulation. What do you mean by double circulation? Blood one circulate from heart to body and body to heart. That is called systemic circulation. Another circulation is the blood goes from heart to lungs and lungs to heart, then goes to body. So heart to lungs, lungs to heart, pulmonary means lung, and heart to body, body to heart is systemic. So since we have a two circulation, twice of blood circulate in the heart, hence it is called double circulation. So this type of question may be asked, what is double circulation? We have a parallel to the blood circulatory system, we have a lymphatic system. So lymph contain lymphocytes which are bigger in cell, that cannot run through the blood. So that creates a problem. So similar to that, trains cannot run, the trains cannot run on the road. Similarly, extracellular fluid, another system need for lymphatic system. So different lymph nodes are there, lymph vessels are there. They also coordinate among themselves. They also exchange the material between them. Then we have a transportation system in plant. So we have a xylem and phloem. Xylems are composed of tracheic vessels, xylem parenchyma, xylem fibers. These elements help the water to transport from lower part to the upper part of the body, unidirectional, from the root to lips, and the lips prepare the food, and the prepared food may sometimes come to the lower part as well as from the lower to higher, where the root store the food, and during the need the root, the food of root may goes to leaf or development of new leaves. So in this way the movement of phloem is bidirectional, while the movement of xylem is unidirectional. So in this way you can say explain easily the transportation of water taking place by transpirational pool, root pressure. When the water enter into the lumen of xylem, you can see this diagram, that this is a root hairs where the water comes in this lumen of xylem from all around. This one you are observing. So when the water come from all around, there is a pressure generated, and that pressure compel the water to rise up. So that is called root pressure. Another, there is a continuous column from root to leaves. So when transpiration takes place, the water vapor evaporate there, that gives a successive transpirational pool, and there is a cohesion force between the water molecules, that pulling effect transfer from molecule to molecule, and ultimately the pulling effect is given that compel the water to rise up. So this is the mechanism. Hardly can ask this question. Absorption of water is introduction of capillary water from root to lumen of xylem, and then goes to upper part of the body. So you can easily describe the even transportation in plants as well as in human beings. So today in this video I think you revise this one. Next time I will also make you the different way. I think if you have any problem, you can communicate with my YouTube channel Royal Education. Okay, thank you.